The timing of this is great for me because this is the first time I've been invited to present this work, and it's just now the final version, I think literally in the last half an hour, being released in Nature. And I want to start by just mentioning the place where I work, which we've just moved to, is a brand new institute in Hampstead in North London, the Institute of Immunity and Transplantation in the Pears building. This is going to be a really state-of-the-art translational immunology research centre. This is Leo Swadling, who led the work in the group and did a fantastic job. The CIC is a UK-wide coronavirus immunology consortium, which I was one of the leaders for, and this really tried to coordinate immunology research across 19 different centres in the UK and funded this work. And the COVID consortium I'll tell you more about was a very innovative healthcare worker sampling collection that we were part of. So I think everyone's aware that despite the highly infectious nature of SARS-CoV-2, some individuals seem to resist overt infection. And of course, many of those will just not be exposed to an infectious dose of the virus. Others are exploring the possibility of genetic resistance. But what I want to talk to you about today is the possibility that some of these people have an early control, or what we've termed abortive infection. So I guess, we've all come across households where everyone except one person has got infected. The data I'm going to show suggests that some of these people may actually just have eliminated the infection in the very earliest stages. So just to summarize, adaptive immunity, you've already heard a very nice presentation about the importance of the early innate immune response, particularly the type 1 interferon response. And then we've been hearing about the role of antibodies and the concern with waning levels of antibodies, which can then be supplemented by booster doses of vaccines, because memory B cells, we and others have shown, do persist even when the neutralizing antibodies have completely waned. And that can bring back up the levels of antibodies. But of course, working hand in hand with those antibodies in any acute viral infection are the T cells, CD4 and CD8 T cells, which both help the antibody response, but also have direct antiviral effects and are highly multi-specific in SARS-CoV-2 and likely to be able to persist longer than antibodies. What was emerging early on in the immunology work on SARS-CoV-2 was that there were a large proportion of people who were tested before the pandemic and found to have T cells that could cross-recognize SARS-CoV-2 in vitro. So we started to wonder whether these T cells, being memory T cells that could then kick into action very quickly, might be able to give the immune system a head start. And then instead of this normal paradigm where the virus comes up and then the antibodies and T cells are induced and bring down and control the virus, if you had pre-existing memory T cells that were able to cross-recognize SARS-CoV-2 due to priming by a similar virus, perhaps these could come up very quickly and actually abort the infection before you had detectable PCR, and perhaps even without the induction of antibodies. And that's what I'm going to show you some evidence for. So to study this, we got involved with this COVID consortium that I mentioned, which was a large study of 731 healthcare workers who were recruited incredibly early, thanks to the foresight of my colleagues who set this up with actually crowdfunding, so that the recruitment was all done during the first week of lockdown in the UK. So obviously with the Wuhan strain of SARS-CoV-2. And what was great about this consortium was not just the early recruitment, but also the thoroughness with which they were sampled. So every week for 16 weeks, these healthcare workers had PCRs, multiple serology tests with Public Health England and neutralizing antibodies. And we could see that there were obviously a highly exposed group, and at that time, the PPE was quite inadequate, and over 20% did get lab confirmed infection. But of course, a large proportion of them remained seronegative. And we did a lot of work early on looking at the duration of immunity and the antibodies and T cells in the lab confirmed group, but now I'm going to focus on this seronegative group. and ask the question, were these all people who were truly unexposed, or could we find some evidence that some of them had actually aborted infection? And so to do this, we had a sub-study where we got a lot more cells from a smaller group of these two. We matched these groups of seronegatives and lab-confirmed infection for all the sort of demographic factors that we could think of, and also for their risk exposures in terms of the type of work they were doing in the hospital. And these blood samples were taken at 16 weeks after recruitment, so looking at the memory T-cell response. And then we also have an unexposed pre-pandemic cohort that we compare these with. Okay, so first of all, just to show you how we defined this seronegative cohort. So as I mentioned, they were very thoroughly sampled over 16 weeks. And from the stage where we were at the peak of the pandemic, so we really should have been picking up exposure, and both with these S1 IgG and NP IgG IgM tests through Public Health England, this group were repeatedly negative. They also had repeatedly negative PCR tests every week for 16 weeks, compared to the lab confirmed group whose PCRs stopped being negative by about week 6 into this study. We also confirmed with neutralizing antibodies. This is a pseudovirus neutralization assay done by Anya McKnight's lab. Again, the seronegative group, all except for 2 with borderline results that we excluded, were negative with pseudovirus neutralization. And I'm not showing here, but Anya also did live virus neutralization at 3 time points in this group, and it was still negative. Then just to also further confirm this, we looked at memory B cells, which we had shown in another study where, as I've mentioned, were able to persist in donors who had lost antibodies. And again, we saw no detectable spike-specific memory B cells in this group, similar levels to what we saw in a pre-pandemic cohort. Okay, so now I'm going to show you 2 pieces of independent evidence that, in fact, a subset of these seronegative healthcare workers appeared to have evidence for abortive infection. So first of all, some T-cell data. So we've been measuring T-cells primarily using the interferon gamma ELISpot, like many groups. It allows us to look at a wide range of T-cell responses, a very rapid reproducible assay. And we were provided with peptides very kindly by my long-standing colleague, Antonio Bertoletti in Singapore, covering different regions of the virus, which I'll come back to talk about in more detail later. But we were using briefly just overlapping pools of peptides, so not predefined epitopes on the whole, predefined overlapping peptides. So like many others, as I've mentioned, have now reported, we also found that a large proportion of our pre-pandemic cohort, so sampled well before the start of this pandemic, had T cells that were able to recognize portions of the virus. So the different colors here represent the different regions of the virus, and then each of these bars is a different donor. But then when we looked at this group of seronegative healthcare workers who had potentially been exposed at work, we saw a stronger T-cell response and a more multi-specific response. And in fact, you can see that that was really comparable to the type of T-cell response that we were seeing in the group, the matched group with lab-confirmed infection. And then you can see this numerically compared here. So here's looking at the overall magnitude of the T-cell response, summing up the T-cells to all the different epitopes that we looked at. The seronegative healthcare workers had significantly more than the pre-pandemic, and no difference really between those and the overt infection, lab-confirmed infection. And similarly with the breadth of the response, so looking at the number of different viral proteins that could be recognized, there was a significant increase in breadth in the seronegative healthcare workers with potential exposure compared to the pre-pandemic cohort, which looked no different really from the lab-confirmed infection. So this is kind of quite a heresy for a lot of immunologists, this idea that you might be getting T cells generated without any detectable antibody, despite looking very thoroughly at multiple time points. But looking at the literature, there are other studies that to some extent support this. First of all, there are a number of studies suggesting that T cell protection can kick in before the antibody response. So in SARS-CoV-2, there's been work suggesting that early T cell responses are really important for the first steps, in fact, of rapid viral clearance in lab confirmed asymptomatic mild COVID. And also with mRNA vaccination, it's been noted that the early protection phase kicks in before the antibodies are detectable and actually correlates instead with early induction of T cells. Similarly, in older studies in flu, it's been shown previously that those with higher T cells were more likely to have undetectable virus by PCR. And in challenge studies, early T cells were able to be detected before the induction of antibody and coincided with viral elimination. And these have been proposed to be due to cross-reactive memory T cells. But then there's also some work from the field that I normally work in. So bloodborne viruses like hepatitis B is what my lab normally concentrates on, but also in hepatitis C, HIV. There were reports many years ago of T cell responses being detectable in highly exposed seronegative individuals and suggested to be attributable to perhaps repetitive low dose exposure. So, but because this concept of T cells being induced without detection of antibody or PCR was quite controversial, we wanted to confirm these data with another approach. And luckily, as part of this COVID-Consortium, we're interacting with many different researchers with different expertise. And Maini Sadeghi, a colleague at UCL, had published this study showing that screening for different blood transcriptional markers as a way of diagnosing SARS-CoV-2, that an interferon-inducible gene, IFNY was a very sensitive and early marker in SARS-CoV-2 with a specificity and sensitivity pretty high, and actually sometimes coming up even before the detection with PCR. And so running this assay with on our samples with Maini Sadeghi, so this is what we found. So first of all, using a pre-pandemic set of samples to set the threshold for detection with mean and 2 standard deviations shown in grey here, there was a small proportion, about 10% of our PCR negative cohort who fell outside that. And then when we stratified these according to their T cell responses and according to the baseline and peak, they stayed flat in the ones without expanded T cells, but those with the expanded T cells tended to have higher levels of this gene, which came up even higher at the peak of follow-up. And this is in the first 5 weeks of sampling. They didn't get such a high induction of the IFI27 gene that we saw in the classical PCR positive ones with lab confirmed infection, but it was clearly significantly higher than in the group without induction of T cells. And you can see again here the longitudinal data. So sampling from weeks 0 to 5 after recruitment into the study, those without expanded T cells stay really nice and flat for IFI27, whereas those with expanded T cells, actually 95% of them at one point or other during this 5 weeks went above the threshold for this interferon-inducible gene. So far, I would argue that our data suggests that on top of this spectrum of SARS-CoV-2 infection that we all know about, ranging from asymptomatic, mild, moderate to severe, and people who are totally unexposed, there's an additional group to add in here with an abortive infection. And so the next question we wanted to ask is there a difference in the T cells that we see in the abortive compared to overt infection? I've shown you that their magnitude and overall breadth is similar, but is there some difference in their specificity? And what I'm going to show you is that, in fact, the T cells in this group of abortive infection preferentially target the virus replication machinery, which is the first viral proteins produced within infected cells. And the focus on this really is thanks to the collaboration with Antonio Bertoletti's group, who early on in this paper, early on in the pandemic, had noticed that there was an increase in non-structural protein T cells coming up in the pre-pandemic samples compared to the infected donors. And so we decided to include not just peptides from the standard structural proteins that many groups are studying, but unusually to also include peptides spanning these regions from ORF1. ORF1 is such a big region that many T cell studies has left it out because it's unmanageable to include it all. But we included overlapping peptides spanning the polymerase, NSP12, the cofactor for polymerase, NSP7, and helicase, NSP13, which together make up what's called the mini replication transcription complex, the core part of the replication machinery of the virus. And as I mentioned, the part that's expressed first in the viral life cycle. So interesting what we see when we look now at the overall T cells targeting the replication transcription complex, RTC, or the structural proteins, is that whereas in green here, the seronegative group have similar levels of structural responses to the overt infection, when you look then instead at the RTC region, this seronegative group have significantly more T cells against this region than both the pre-pandemic group and the group with a classical overt infection. And then when we look in more detail, we see that there's a significant enrichment for NSP7 and NSP12, particularly responses in this seronegative group compared to the seropositive classical infection group. And breaking this down further into the different subpools that we use, because NSP12 in particular is a very big region, we saw that for all of these subgroups, there was a significantly higher response present at 16 weeks in the memory T cell response in those with seronegative compared to overt infection. So are these T cells being induced de novo, or are they being boosted from pre-existing cross-reactive responses? Well, to get at this question a bit further, we teamed up with our colleagues in the computational UCL genetics department, Francois Bellew, Lucy Van Dorp, and Cedric Tan. And they did some lovely bioinformatic analysis on the degree of conservation between SARS-CoV-2 and the 4 seasonal common cold human coronaviruses, which everyone has been exposed to, particularly healthcare workers that we were studying are widely exposed to these viruses. And interestingly, what they found was that the replication transcription complex that we had started noticing looked interesting in terms of T cells was also very interesting in terms of conservation, particularly around NSP12 and 13. There was high levels of conservation sequence identity between SARS-CoV-2 and these seasonal coronaviruses. And that's summarized here with the degree of conservation plotted for these RTC proteins being significantly higher than all the structural proteins. And actually, they've also shown, although I've not got that in the diagram, they've now published actually that there's also this same degree of high conservation in this region with all other animal coronaviruses. And they went on to take a look at all the SARS-CoV-2 variants that are circulating. So this was a subsample of the GISAID sequences available in July 2021. And again showed that there's actually less diversity amongst all these variants in these regions, particularly NSP12 polymerase and NSP13, than most of the other regions of the virus. So there's a region of the virus which is expressed early, highly conserved in pre-pandemic samples, both from London and from a cohort from Antonio in Singapore. The NSP12 region polymerase was the one that was most frequently recognized by samples taken from before the pandemic. So, so it seems like this is a very common region to be targeted by T cells, which are probably cross-reactive with, for example, seasonal coronaviruses. And then this region is, again, most commonly targeted in the seronegative uninfected healthcare workers. So these pie charts indicate the degree of people with a response, and then this is the the strength of the response. So both the strength and the number of people responding is highest for NSP12. I haven't really got time to go into the details of this, but Leo did a lot of elegant work then showing that in vitro there was cross-reactivity between T cells that could both recognize SARS-CoV-2 and some of the common cold coronaviruses as a likely potential source, not probably the only source, but one potential source for cross-reactivity. So now we really wanted to say, okay, there are these pre-existing T cells that are targeted to these conserved regions. Can we find evidence that are actually responding to SARS-CoV-2 in vivo? And here's the data which we managed to get because we were able to go back to very precious samples that have been taken just at the time of recruitment. So, and then
giving you the 2-minute mark.
Okay, and then compare those with the memory responses, and we could see that in many cases there was evidence for expansion of polymerase NSP12 specific responses. And this is summarized here for another cohort where again NSP12 is significantly expanded after exposure. And very importantly, when we then went back to the baseline samples, these NSP12 specific responses were significantly enriched in those who went on to have abortive infection compared to those who had a classical lab confirmed infection. So this just comes back to the summary cartoon to end up with evidence that an early T cell response it may be able to abort infection and prevent PCR being positive and antibodies being induced. We've got lots of unanswered questions. There's a possibility that there's some sequestered mucosal antibodies that we haven't looked into. Of course, in human studies, we don't know whether these RTC-specific T cells are really causal or associative. It's possible that an innate mechanism such as RIG-I may be contributing, and we really want to do some animal studies, human challenge and vaccine studies to look into this further, find out how common this is in bigger cohorts, whether there's an HLA association, age effects, geographic effects. What's the durability of these type of T cells and can they influence the response to vaccines and can they protect against more infectious variants? But overall, we provided 2 pieces of independent evidence for abortive infection. We show that these pre-existing T cells commonly target the RTC that's highly conserved amongst coronavirus and variants, and they're selectively enriched before and after exposure in those with abortive infection. So we think they may be particularly effective at early control and long-lived pan-coronavirus reactivity. And there's really a strong rationale for thinking about including these proteins to supplement spike in next generation vaccines. Okay, and I just want to finish thanking everybody again, thanking Leo for leading the work, other people in my group who paid a vital contribution, thanks to the whole Maini lab, to the big COVID Consortium for absolutely vital work in collecting these samples and collaborating with us. And I've mentioned many other collaborators along the way and our funders. Thank you.
Thank you. Yes, we can open now for questions. I have a question from Dr. Duffy.
Hi, thanks, Mala. It's a beautiful study. Have you been able to look in children to see if these polymerase-specific T cells might be more present and potentially explaining why they're less affected given how they're regularly exposed to colds?
No, we haven't. We'd love to do that. I mean, there is evidence for more cross-reactive antibodies in kids emerging, but it'd be fascinating to see if that's complemented by T cells. We haven't had a chance to do that yet. Okay, thanks a lot.
Thank you. I don't see any additional questions. Oh yeah, so Leah van der Hoek.
Yeah, hello. Thanks for a great talk. I would love to propose to you that it would be nice to look at these T cells also after an acute infection by one of these seasonal viruses. So if you know that these antibodies rise, which I will present in the next presentation, but it would be nice to also look then at the T cells, which we have, I must say, and then look what is happening, what kind of T cells are activated when you get these common cold coronavirus infections.
Yes, and now we're starting to be able to get those samples again, because of course, I mean, one of the nice things about this study was that at that time there were no other viruses circulating. And we did look at coronavirus, seasonal coronavirus serology in this group. And it was positive across the board, as you would expect in healthcare workers, but it wasn't increased in the group who had these expanded T cells. There was no evidence that they'd actually had a coincident seasonal coronavirus, as you would expect, because there just wasn't anything much else apart from SARS-CoV-2 circulating at the time. But now, of course, we're starting to get other viruses coming back. It would be really interesting to see. I agree.